सो आप सभी को बहुत बहुत स्वागत है। आज हम पढ़ने वाले हैं फार्माकोलॉजी का चैप्टर नंबर 1। ये गुडमैन गिलमैन बुक से है। द फार्माकोलॉजिकल बेसिस ऑफ थेरापेटिक 13 एडिशन वाला है। चैप्टर नंबर 1, ड्रग इन्वेंशन एंड फार्मास्यूटिकल इंडस्ट्री। ओके, तो आज हम इसे डिटेल में देखेंगे। ये थोड़ा बहुत मैंने रिकॉर्डिंग किया, उसके बाद डिलीट किया। चलो कोई बात नहीं, स्टार्टिंग से आज पढ़ते हैं। सो चैप्टर का नाम अगेन, चैप्टर नंबर 1, ड्रग इन्वेंशन एंड फार्मास्यूटिकल इंडस्ट्री। ओके, द फर्स्ट एडिशन ऑफ गुडमैन एंड गिलमैन published in 1941 helped to organize the field of pharmacology, giving it intellectual validity and an academic identity. The edition begins, the subject of pharmacology is a broad one and embraces the knowledge of the source, physical and chemical properties, compounding, physiological action, absorption, fate and excretion, and therapeutic uses of a drug. A drug may be broadly defined as a chemical agent that affects living protoplasm, and few substances would escape inclusion by this definition. This general principle section provides the underpinning for this definition by exploring the process of a drug invention, development, regulation, followed by the basic properties of the interaction between the drug and biological system, pharmacodynamic, pharmacokinetic, including drug transport and metabolism, and pharmacogenomics, with a brief foray into drugs toxicity and poisoning. Subsequent section deals with the use of a drug as a therapeutic agent in human subject. Use of the term invention to describe the process by which a new drug is identified and brought to medical practice, rather than the more conventional term discovery, is intentional. Today, useful drugs are rarely discovered, hiding somewhere waiting to be found. The term invention emphasizes the process by which the drug are sculpted and brought into being based on experimentation and optimization of many independent properties. There is a little serendipity for early experience with plants to modern chemistry. The human fascination and sometimes infatuation with chemicals that alter biological function in ancient and result from long experience with and dependence on plant. Because most of the plant are root bound, many of them produce harmful compounds for defense that animals have learned to avoid and human to exploit or abuse. Earlier edition of this text describe example the appreciation of a coffee मतलब caffeine by the prior of an Arabian convent who noted the behavior of goats that gambled and frisked through the night after eating the berries of the coffee plant. The use of a mushroom and the deadly nightshade plant by professional poisoner of belladonna मतलब beautiful lady to dilate people of the Chinese horn ma huang containing ephedrine as a circulatory stimulant of curare by South American Indian to paralyze and kill animals hunted for food, and of poppy juice, that is opium, containing morphine from Greek Morpheus मतलब the god of dreams for pain relief and control of dysentery. Morphine, of course, has well known addicting properties mimicked in some ways by other problematic मतलब recreational natural product nicotine, cocaine, ethanol. Although terrestrial and marine organisms remain valuable sources of a compound with pharmacological activities, drug invention became more allied with synthetic organic chemistry as that discipline flourished over the past 150 years. Beginning in the dye industry, dyes are colored compound with selective affinity for biological tissue. Study of these interactions stimulated Paul Ehrlich to postulate the existence of a chemical receptor in the tissue that interact with and fixed the dyes. Similarly, Ehrlich thought that unique receptor on microorganism or parasite might react specifically with certain dyes, and that such selectivity could spare normal tissue. Ehrlich's work culminated culminated in the invention of ars phenamine in 1907, which was patented as salvarsan, suggestive of the hope that the chemical would be salvation of humankind. This and other organic arsenical were used for the chemotherapy of syphilis until the discovery of penicillin. The work of Gerhard Domag demonstrated that another dye, prontosil, the first clinically useful sulfonamide, was dramatically effective in treating streptococcal infection, launching the era of antimicrobial chemotherapy. The collaboration of pharmacology with chemistry on the one hand and with clinical medicine on the other has been a major contributor to the effective treatment of a disease, especially since the middle of the 20th century. Okay, source of a drug. Small molecules are the tradition. With the exception of a few naturally occurring hormone, example insulin, most drugs were small organic molecule, typically less than 500 Dalton, until until इसका पेज नंबर भाई क्या है, कुछ मेंशन नहीं. until recombinant DNA technology permitted synthesis of a protein by various organisms मतलब bacteria, yeast and mammalian cell. The usual approach to invention of a small molecule drug is to screen a collection of chemicals मतलब library for compound with the desired feature. An alternative is to synthesize and focus on close chemical relatives of a substance known to participate in a biological reaction of interest. Example, congener of a specific enzyme substrate chosen to be possible inhibitor of the enzymatic reaction, a particularly important strategy in the discovery of anti-cancer drug. Drug discovery in the past often resulted from serendipitous observation of the effect of a plant extract or the individual chemical on animal or human. Today's approach relies more on high throughput screening of libraries containing hundreds of thousands or even millions of compounds for their capacity to interact with a specific molecular target or elicit a specific biological response. Ideally, the target molecule are of human origin, obtained by transcription and translation of the cloned human gene. The potential drug that are identified in the screen hits are thus known to react with the human protein and not just with its relative मतलब ortholog obtained from the mouse or another species. Among the variables considered in screening are the druggability of the target and the stringency of the screen in terms of the concentration of a compound that are tested. Druggability refers to the ease with which the function of a target can be altered in the desired fashion by a small organic molecule. If the protein target has a well-defined binding site for a small molecule, example catalytic or allosteric site, chances are excellent that hits will be obtained. If the goal is to employ a small molecule to mimic or disrupt the interaction between two proteins, the challenge is much greater. From hits to lead. Initial hit in a screen are rarely marketable drug, often having modest modest affinity for the target and lacking the desired specificity and pharmacological properties. Medicinal chemistry synthesize derivative of the hits, thereby defining the structure activity relationship and optimizing parameters such as affinity for the target, agonist antagonist activity, permeability across cell membrane, absorption and distribution in the body, metabolism and unwanted effect. This approach this approach was driven largely by instinct and trial and error in the past. Modern drug development frequently takes advantages of a determination of a high resolution structure of a putative drug bound to its target. X-ray crystallography offers the most detailed structural information if the target protein can be crystallized with the lead drug bound to it. Using techniques of molecular modeling and computational chemistry, the structure provides the chemistry with the information about substitution likely to improve the fit of the drug with the target and thus enhance the affinity of the drug for its target. Nuclear magnetic resonance NMR studies of the drug receptor complex also can provide useful information, albeit usually at lower resolution, with the advantages that the complex need not to be crystallized. The holy grails of this approach to drug invention is to achieve success entirely through computation. Imagine a database containing detailed chemical information about millions of chemical and a second database containing detailed structural information about all human proteins. The computational approach is to roll all the chemical over the protein of interest to find those with high affinity interaction. The dream becomes bolder if we acquire the ability to roll the chemicals that bind to the target of interest overall other human protein to discard compound that have unwanted interaction. Finally, we also will want to predict the structural and functional consequences of a drug binding to its target, a huge challenge as well as all relevant pharmacokinetic properties of the molecule of interest. Indeed, computational approaches have suggested new uses for old drug and offered explanation for recent failure of a drug in the latter stage of clinical development. Large molecular increasingly important. Okay. Large molecule are increasingly important. Protein therapeutics were uncommon before the advent of a recombinant DNA technology. Insulin was introduced into clinical medicine for the treatment of diabetes following the experiment of Banting and Best in 1921. Insulin purified from porcine and bovine pancreas are active in human, although antibodies to the foreign protein are occasionally problematic. Growth hormone used to treat pituitary dwarfism exhibit more stringent species specificity. Only the human hormone could be used after purification from pituitary gland harvested during autopsy and and such use had its danger. Some patients who received the human hormone developed uh, Creutzfeldt Jakob disease, the human equivalent of a mad cow disease. Mad cow disease, a fatal degenerative neurological disease caused by prion protein that contaminated the drug preparation. Thanks to gene cloning and the production of large quantities of protein by expressing the cloned gene in bacteria or eukaryotic cell. Protein therapeutics now use highly purified preparation of a humanized protein. Rare protein can be produced in quantity and immunological reaction are minimized. Protein can be designed, customized, and optimized using genetic engineering techniques. Other types of macromolecules may also be used therapeutically. For example, antisense oligonucleotide are used to block gene transcription or translation, as are siRNA. Protein used therapeutically includes hormone growth factor, example erythropoietin, granulocyte, colony stimulating factor, cytokine, and a number of monoclonal antibodies used in the treatment of cancer and autoimmune disease. चैप्टर नंबर 34, 36 एंड 67 में इसके बारे में देखेंगे. Murine monoclonal antibodies can be humanized by substituting humans for mouse amino acid sequence. Alternatively, mice have been engineered by replacement of a critical mouse gene with their human equivalent, such that they make completely human antibodies. Protein therapeutics are administered parenterally and their receptor or target must be accessible extracellularly. तो आज के लिए इतना ही. यहाँ पर हम इसको एंड करेंगे. आज का डेट कितना? 29 January 6. Thanks for watching.